Now lastly, the most important one is mitochondria. Mitochondria use molecular oxygen to, in effect, burn sugar and small fatty acid molecules to produce adenosine triphosphate, ATP, which is used by all energy-requiring cellular reactions. Glucose is first converted to pyruvate in the cytosol by glycolysis, producing in the process a small net amount of ATP and reduced nicotinic adenine dinucleotide, NADH. Glycolysis does not require oxygen, so when oxygen is limited, this anaerobic respiration can supply some ATP, with NADH being reoxidized to NAD+ by the metabolism of the pyruvate to lactate. However, under normal conditions where there is sufficient oxygen, oxidative phosphorylation in the mitochondria produces 15-fold more ATP for each glucose molecule than does glycolysis. Pyruvate and fatty acids transported into the mitochondrial matrix act as substrates for enzymes that drive the citric acid, Krebs, cycle, which generates NADH and the waste product carbon dioxide. The electron transport chain, a series of enzymes in the inner mitochondrial membrane, then uses molecular oxygen to reoxidize NADH to NAD+. In doing so, it generates a hydrogen ion gradient across the inner membrane which drives the ATP synthase. The Krebs cycle takes place inside the mitochondria. The reaction is catalyzed by succinyl-CoA synthetase, which produces guanosine triphosphate GTP, which is structurally like ADP. The enzyme nucleoside diphosphate kinase catalyzes the conversion of GTP to ATP in the intermembrane space. Did you know that the inner membrane of the mitochondrion is impermeable to ATP? A protein complex known as the ATP-ADP translocase is needed for the export of ATP in return for the import of ADP and phosphate anion. Now if we look closely in this step of Krebs cycle, succinyl-CoA undergoes an energy-conserving reaction in which succinyl-CoA is cleaved to form succinate. This reaction is accompanied by the phosphorylation of guanosine diphosphate GDP to guanosine triphosphate GTP. The GTP thus formed then readily transfers its terminal phosphate group to ADP, forming an ATP molecule. The reaction is catalyzed by the enzyme succinyl-CoA synthase. In summary, ATP is readily available to release its energy rapidly wherever it is needed in the cell. To replace ATP used by the cell, much slower chemical reactions break down carbohydrates, fats, and proteins and use the energy derived from these processes to form new ATP. More than 95% of this ATP is formed in the mitochondria, which is why the mitochondria are called the powerhouses of the cell. That was all about the cell functions related to physiology. We have more videos coming up related to the cell, so stay tuned.
Watch our medical videos anytime and anywhere. Download sqadia.com app now.